Okay, this is lecture 16, and today we're going to look at models of sequence evolution. This is a bridging lecture in some sense from the Markov models we built the last couple lectures in the context of gene finding and in binding site analysis, and we're going to move that towards phylogeny and sequence similarity and alignment. So we're going to build on the Markov models to look at how we can use those to model nucleic acid evolution and amino acid level models. And we're going to get into a little bit about evolutionary details there with how selection can be modeled, etc. And I'm going to show you the different kinds of genetic events that can occur, both in healthy individuals and across species, everything from the whole tree of life, really. And then I'll end by talking about its relationship to cancer. Now, I put some points of reflection here that I'll leave those to for you to come back to at the end. I don't think it's worth going into them here. We're going to switch over to some lecture notes now. See you in a second. All right. Markov models of evolution. We've already seen Markov models now, a couple classes, and we know that there are three components. There's the network, nodes and arcs between them, and the probability of transiting from one arc to the other. And a third thing, which is the initial probabilities, pi A, pi C, etc., that tell us, that describe with which probability in which state we start. Now, Markov models actually lie at the heart of classical statistical genetics, and I doubt that many of you have actually taken a statistical genetics course by this point, which is really unfortunate. Statistical genetics underlies so many different areas of research, like epidemiology, and I should say not just research, health policy, health science, clinical practice. For example, epidemiology is the study of disease in the population. So diseases that have a genetic component are modeled using statistical genetics. It describes how Mendelian traits evolve. It describes how, as a species, we evolve. All sorts of different angles on evolution, basically. So usually what these models for sequence evolution are referred to as substitution models in genetics. And but there's also a lot of computational biology contexts where we also need Markov models that capture how sequences evolve, and that's going to be the subject of the end of today's lecture and next lecture, where we look at phylogeny, molecular evolution, and sequence alignment. Okay, so the basic principle here is that we're going to, instead of walking along a chromosome, remember that we've always been kind of walking from left to right, say, across the chromosome and asking, is this a binding site? Is this a gene? Here, conceptually, we're going to watch a site. So here, at the beginning of the lecture, a site is just one nucleotide in a genome. So here I've drawn one, and then there's like any number of nucleotide sites between, then a T, and then some more nucleotides. I've just highlighted 4 of them. Okay, and along this axis here, I'm going to call it time, but that's going to be the trickiest concept today, is what do I really mean by time? And that's where you're going to have to really go slow. All right, so the Markov model here is going to, we want to focus on it on one site over time. Okay, so now the first site here, a C, the idea is that I started in the state C, and I'm going to watch that, I'm going to stare at that site over, years or generations or eons or millennia or whatever, and I'm going to watch it, and after this amount of time, it's still a C. And if I watch it some more, it's still a C. And if I watch it some more, it's still a C. So that's pretty boring. What we can see, though, in the second site is that during this evolution between this point and this point, there's been a point mutation. We can call that a point mutation. So T has flipped to A. And then the rest of the time, it's every time we've looked at it at these different time points, we see that it's an A. We could see, for example, here that you could get multiple hits at the same site. For example, here, T, then G, and then C, and then A. So it's covered the complete spectrum of nucleotides. And finally, you can have what's called homoplasy, where a site mutates back to what it began with. So here, G evolved to an A, and then we see it as an A, and then it flips back to a G. Okay? All right. So I think that's pretty simple so far. Okay, so what I want to say here is that each site is modeled by a Markov model. Okay, so we can think of that Markov model as controlling this site, right? Okay, it's generating this site. So we're bouncing through this Markov model over time, and we're outputting whatever nucleotide we want. So we don't need a hidden Markov model here. We just think of this as a pure Markov model. Okay, so that's important. Now each of these, in the simplest models that we're going to look at today, each of these Markov models are independent, but they're identical. Okay, so this one evolves independent of this one, but the parameters for this one, including the start probabilities, okay, they're the same for all of these different sites. So the sites are all described by the same model, but the models evolve independently. In other words, this one here, I started in a C, and I ended up in a C. And in this one here, I started in a T, and my evolution started there, proceeded from there, sorry, a T, and this one started in state G. So they may all have the same parameters, but the evolution occurs independently. And there's a term for that called IID that you see quite often in the literature, and it means identically independently distributed. Okay, so it's exactly that, IID. So, well, what do we mean by time, really? Okay, and this is going to be, like I said now a couple times, this is the trickiest thing of today's lecture. Okay, so, well, let's start with something really simple. Let's go back to because we're all experts in Markov models, let's think about a step in time to be one step in the Markov model. So we could think of it as being like, you start off here in state C, okay, at the beginning of time, and one tick of the clock, maybe that's a second, or a year, or 1,000 years, or 1,000,000 years, whatever you want one step to be, this C, you roll the dice, and in this case here, C stays a C, okay? All right, and then that gets us to this point here, at step time 1, right? And then whatever your time step is, okay, you wait that, in a sense, you can wait. Like if it's 1,000 years, you imagine that you roll forward the clock 1,000 years, and you roll the dice again. In this case here, you switch to a T, and now at this time point, you're in state T. And then again, you make one step in the Markov model, and that case here, it's a self loop, and you stay here in state 3. Now, okay, so let's get that point straight. I mean, I can use this Markov model in an abstract way to think of it as a discrete time points, okay? And I could somehow synchronize those discrete time points with a allotted time point. It doesn't need to be 1 second or 1 hour or 1 minute. Each step could be, let's say, 1,000 years. Maybe that's a good idea. Maybe it's not. I'm not sure. But we can kind of think as of dividing our time interval up into equal-sized segments. And if this time interval is 4,000 years or 3,000 years, sorry, this is 1,000 years, this is 1,000 years, and this is 1,000 years. Okay, so just try to keep that in mind. Now, our transition matrix here that parameterizes this graph looks like this, where the probability of A going to A A going to C, that would be this arc, A going to G, that, and A to T, etc. This is the same as before, but we would call this our transition matrix, right? Our probability matrix. And you read this as the probability that nucleotide X changes to Y in exactly one step, all right? But so far, nothing has changed from what we talked about before with Markov models, except now I'm saying, like, let's kind of think about time in this kind of discrete stepwise way, all right? So what is the relationship between time, and I don't mean in a metaphysical sense, just as we know it sort of thing, and one step in the Markov model? Let's look at this a bit more closely. Now, I warn you, it might be a little bit difficult to understand some steps here, but try to stay with me, okay, and ask questions if you don't understand. So think of, maybe go back to your calculus days and think of a step in the Markov chain as being an infinitesimally small unit of time. We can call it delta t. That's, maybe I should even write that right. Just, I mean, I always found it a bit difficult to remember these Latin and Greek letters. So that's delta. Okay, so let me just get rid of that for now. I'm trying to keep the notation down here, but I need a little bit. So let E be my alphabet of nucleotides, A, C, G, and T. So that's the possible states, right? Okay, I think we're all comfortable with that. Let's let P at T be this vector. Well, it just means that we have a probability for A, a probability for G, for C, and T. You guys know that. It's like a vector numeric in R. It's gonna, each one of those values is the probability of being in that state at that time. So in our previous notation, this is basically, sorry, I gotta change my color here. This is basically our vector pi, okay? So we have pi A, pi C, pi G, and pi T, right? Nothing new there. I mean, you guys are comfortable with that. So all I'm going to add there is that I just want to ask that you think about this a little bit maybe differently than what we thought of before as a starting probability. Rather, imagine that you're looking at one site in the Markov chain in your head, okay? So we go back here and maybe you're staring at this site here, okay? And I imagine that you sort of simulate without looking at the site now, okay? You let it sort of run the Markov model for 1 million years, okay, or 1 billion years, for a long, long time. And then you look at it. And the question would be, what's the probability of observing an A or a G or a C or a T? I think that basically it might be something like the probability or the frequency of each nucleotide in the genome, okay? I hope that's kind of clear. So again, the thought experiment here is that you imagine that you're going to look at the, let's say, the 99th nucleotide in the yeast genome. Now, you don't look at it first. You're just saying, okay, I'm going to look at it. And then, because you're godlike, you can then allow evolution to run for 1 billion years. And then you look at that 99th site in the yeast genome. And, well, it might be, these probabilities might be well modeled by the background frequencies of the nucleotides. So if C and G are, let's say, total 60% probability, so 30-30, and A and T are 20 and 20, then the probability that you're in state G after 1 billion years of evolution would probably be about 30%, right? Okay. It'll come clear over time. But recall that these guys, this is basically our vector. What's going on with my pen? Sorry. This is basically our vector pi that we all know and love from the previous lectures. Now here, mu sub xy, I should make sure you remember that, mu sub xy, just is the transition rate from state x to state y. That's nothing new, right? That's just basically, for example, mu of a to c, that's mu sub ac, right? And this would be mu sub ta, right? So that's this arc here, and mu ca is this arc here, right? So these are just your transition probabilities as before, nothing new. This is new, and you need to make sure you understand it. Let's let mu sub x be the probability that I transition away from x, okay? So I can write that as the sum of all the nucleotides not equal to x, the mu of x going to y. Okay, so what this is graphically, it's easy to see, is we're saying we're at A, we want mu sub A, and mu sub A is going to be this probability plus this probability plus this probability, mutating away from A, but not this guy here. We remove that guy from the game. So it's the probability of leaving, right? And that's exactly what I'm writing there. I can write this out more explicitly for you. So if I was, say, in nucleotide A, nucleotide A, and I wanted to know mu sub A, then that would be mu sub, the sum of mu sub Y, mu sub x to y, right? Which is going to be mu sub c plus mu sub g plus mu sub t, right? Okay? All right. So it's the change in probability. Okay, so now it's going to get a little bit complicated, perhaps, for some. So the change in probability over time, over a small period of time, this delta t, we can write it as follows. For A, let's look at nucleotide A. So the probability I'm at state A at time t plus delta t. Now what is that? Let's go back and draw that out because I want to go very slow with this. So this is my time, and this is time t. And so you could think of this as being a little bit of an interval, a small short interval is t plus delta t, right? Okay, it's my step forward. So the probability I'm in state A at t plus delta t, I'm in state A here. Well, I'm going to use a different color. Let's see here. What's a nice color? Purple. What's the probability that I'm in state A at time t plus delta t? That's what this equation is here. Now it looks complicated, but it actually is quite simple. Well, it's the probability at time t I was in state A. So the hypothetical here is that I was in state A at time t. Okay, and now I subtract off the probability that I leave state A, right? So between this point and this point, I might have left state A, right? I might have mutated away, and that's equal to the probability that I was in state A at time t times the probability, the event that occurs, it's multiplication, that this mu sub A over delta t, okay, over that time interval, right? So if you think of that then as delta t as being one step in the Markov chain for simplicity, it's like saying I'm in state t and in one time interval I leave, right? And that's the probability of mu sub A, which was the sum, again, that's this guy here, right? That's mu sub A, remember, the probability of mutating away. Okay, so now the other thing to consider here, well, that's negative, right? Those are not, that's the events where I'm not in state A at time t plus delta t, right? It's the events where I walked away. So that's why I'm subtracting here. I'm subtracting because I'm not in state A at time t plus delta t. But I add these guys because they're examples of where I am. And so basically these now here are that for all nucleotides not A, right? So this would be C, G, and T. I need to sum the probability that I was in that state at time T, but then I transited to, I left state A, I left state Y, and I entered state A over that small time period, right? I sum these because there's 3 different independent events. Either Y could be C, G, or T. And that the probability of C, G, or T is described by this part. And then this is the probability that I went from C or G or T back to A or to A. And this is over that time interval there. Okay. Now, so graphically, the first case basically is that I'm state A and I leave it, so they're not and I leave 3 different ways. That's not good, so I subtract them off. The other possibility is that at time T, I'm somewhere else, and there's 3 such cases, and over that time point, I get back to state A, right? Okay. All right. So here I've just copied that from above, so we have that on hand. And actually, this kind of can be simplified as writing the probability I'm in some state at time t plus delta t is the probability that I'm in that state at time t, plus the probability that I'm at state times this matrix Q over the time interval, and this Q relates to these equations here. That's a little bit abstract, but I just and I don't, I haven't gone through every tiny step there, but I want you to understand that often in genetics you're given what is called a rate matrix, and that's different than the probability matrix that we've been talking about. It comes from the Markov chain, so but it's a little bit different. It's part of the equation. And so here, in this model here, we would write it as minus mu A, oops, wrong mode, minus mu A, and then you see here it's A to G, A to C, A to T, G to A, C to A, T to A, and along the diagonal you'll see it's always the negative mu. And that's just the way of writing this expression, this matrix, so that it fits this equation here. Okay, so that's what you need from your matrix Q. It has to be of this form. Okay, and often these statistical genetics models for sequence evolution don't give you the probability matrix that you'd expect from a Markov chain. Instead, they give you this intermediate product called Q. Okay, so I'm sure that's quite abstract right now, and some of you are asking, like, where's this going, and how does that really work? Like, I'm not really, that there's a lot of probably a lot of questions. Okay, so let's go back a little bit and work through this. Here's an example where now I'm looking at the probability that I'm in state C at time t plus delta t. Okay, so we could think of this as being time t, and then, well, some number of steps through the Markov chain, and we're at time t plus delta t. All right, so delta t is not equal to one step in the Markov model, right? We have to be careful about that. It's an evolutionary time. We can think of it being very small, but it might correspond to many steps, right? Here's the first step, here's the second step, there. Now, let's go through this, right? What could happen? I'm in state C, right? And I stay in state C the whole time. I could just be walking through that Markov model, and I'm just continually taking that self loop, right? That would be the equivalent of, I'm just staying the whole time in state C by taking that self loop, right? Another possibility is that I go to state A, whatever, and then I go to a bunch of other states, and by dumb luck, I end up back at state C at that time. What I'm not interested in is starting in state C and bouncing around and not ending up in state C, right? That's not what I want. I want this, the probability that I do end up in state C. And so the claim is that this is equal to the probability that I'm in state C at time t plus the probability that I'm in state C at time t times this magic matrix, this rate matrix Q times delta t. And that's kind of pretty advanced continuous time Markov models of where that equation comes from, but it's following the logic back here, this example here that I was describing, right? Of that argument of what can happen, right? And that matrix Q has really nice properties from mathematics, so that basically it's summarizing all the possible things that could occur between this state here and this state. So you could think of enumerating all possible ways of walking through that chain. I can't, in a four-state, in a four-state model, that's not too hard, right? You could say CCCC, CCAC, CCTC, CCGC, right? And then, then you would, another one would be CCACC, well, it depends how you want to count now, but let's say AC, CAGC, right? So, well, you're getting a lot of possibilities, right? And essentially there's 4 possibilities for this position. There's 4 possibilities for this third position. There's only 1 possibility for the N1 and 1 possibility for the first one. There's 4 possibilities for the third, and there's 4 possibilities for this one. And so in total there is going to be, if I was to write them all out, 1 times 4 times 4, so there's 16 different paths. Now the nice thing about that matrix Q is that it basically has these special properties that it takes all those paths into consideration. Sorry, that was my dog. Okay. So that leads to the first of these very classic models for nucleic acid evolution, and that's the Jukes-Cantor model from 1969. Okay. So it assumes all base, it's very simple, it's not very realistic at all, but it assumes all base frequencies are equally likely, A, C, G, and T, right, are all the same. So we can think of them in our old notation of pi A, pi C, pi G, etc., or in the mutation of the previous slide of, or a few slides ago, these probabilities back here. Either notation's fine. Okay, so we assume they're all 1/4, all right? And there's only one parameter here, and that's going to take a little bit of time to describe, but it basically is controlling the rate of mutation. Okay. So let me try to give you some simple intuition about how Jukes-Cantor and all these models effectively work. So we have our Markov model here, and let's suppose, for example, to start with, that we have that everybody's equal at 1/4, and even this self arc of 1/4. Okay, and well, what I'm going to try to get here is to establish here is that having everybody equally likely, including the self arc, is a really high rate of mutation. It's not realistic, but I'll show you why. So suppose we start in state A. I'm picking on A today. Okay, so what is the probability that I mutate away after 1 step? Remember, that's mu sub A. It's the probability that I go to either A, G, C, or T and not A. And so after 1 step, the probability is 3/4, right? So after 1 step of our Markov chain, 1 unit of evolution, in essence, more than half of the sites will be mutated, right? I mean, 3/4 are likely to be mutated. So that doesn't seem very realistic, right? I mean, our genomes don't evolve that fast. Between all of us in this room, we have about, between any pair of individuals in this room, we might have, let's say, 1% of our nucleotides that are different, but, 1% out of 3.6 billion base pairs is very small, right? And some of us maybe are closer to being siblings than others, but we probably all have a few generations between us of humans, right? And so it's not the case that 3/4 of our genomes are different, right? So what about 2 steps? Well, how would I calculate that? Well, that goes back to that Q matrix, but here's when you do it by hand. Here's what can happen after 2. If I started in state A, I could stay in A, and then I could mutate away. There's 3 ways to mutate away to G, C, or T, so it's 1/4 times 3/4. A different thing that could occur, a different series of events, that's why it's plus, is that I stay in state A, I mutate away from state A, and then whatever state I'm in, so that occurs again with 3/4, because I have either go to G, C, or T, each with 1/4, right? Now, if I'm in state G, for example, what's the probability I don't go back to A? Well, it's either I stay in G, I go to T, or I go to C, and so that's 3/4. So that's 3/4 times 3/4. So this, when you add it all up, is 3 over 16 plus 9 over 16. That's 12 over 16. So it's actually 3/4 still. So again, after 2 steps, there's been an awful lot of mutations. So yeah, that's what I've annotated here. I stay, then leave, and I leave, then I stay away. 3 steps, well, it gets a bit longer. I won't go through this, but that's a good exercise there. You might want to stop the video and just ask yourself if you can understand what I'm writing out there, and hopefully I haven't made a mistake. So I guess my point here is that even when there are a few steps in the in the Markov model, you get a lot of mutations, right? Too many, our genomes don't evolve so quickly, and so we need to control the rate of whoops, I'm sorry, it's not quite coming over. We need to control the rate of mutation. That's the trick, and that's what Jukes-Cantor 69 gives us. It's a way of basically inflating the probability that you stay the same nucleotide. So these self-arcs are very important. So then this is the Jukes-Cantor model. I'm going to take one little more baby step to get this, make it as simple as possible. I really want this function f at mu. I don't know what that is yet, but I want this function f at mu so I can subtract it off the minus 1/4. And it's the same f at mu everywhere, right? All over the place, right? Except the diagonal. I'm not going to worry about the diagonal right now because I know that this entry here is 1 minus all of these guys, right? I mean, I can write that out actually. That's going to be 1 minus 3 over 1/4, right? Because it's always 1/4. Well, I'm going to 3 1/4 minus f at mu, right? So I don't need to fill in that that entry on the diagonal. It's easy to figure out what that should be because it's determined by the other guys. Now, if this f at mu, if I choose f at mu to be close to 0, then this is going to approach 1/4, right? Everywhere. And so that's going to make this a also to be 1/4, right? So it's exactly the example of the previous page. And that's going to mean that there's a very high rate of evolution, right? There's a lot of mutations at every site. And if you let evolution run, simulate evolution for even a small amount of time, which is not realistic. But if I make f at mu to be close to 1/4, so the closer it gets to 1/4, the closer these values become 0, which means this A approaches 1, which is kind of more realistic, right? That most of the time you just stay in state A, right? It's a very rare event that you mutate away from A. Okay, so I won't derive where this equation comes from, but Jukes-Cantor suggested this equation to describe PIJ. Sorry, I should have put this here. Oops, this is PIJ. This is matrix P. And so the ith, jth entry of P, okay, with respect to mu, right? This is our parameter to that function now. That's going to be equal to 1/4 minus 1/4 e to the minus 4 mu over 3. Again, I won't explain where that comes from, but that's for all the non-diagonals, right? So this here, this component here is my f at mu, okay, for all non-diagonal entries. And my diagonal entries, not too surprisingly here, you have this 3/4. It's the same thing that's coming in there. And the rest of it looks the same, right? Okay, if I'm on the diagonal where A is equal to A, C is equal to C, G, and T, okay? Now, this is important now, that mu is going to be the number of expected changes per site. So I really would like you to stop the video right now and just think about that for a little bit. You might think about it as that you have 100 nucleotides in a row, okay? Back to the, let me draw this. You might think of, if you have 100 for, think of it in different ways. So one way is to think of 100 nucleotides, and what is the expected number of changes across those 100 nucleotides over your period of evolution, okay? So if you're gonna if you're playing God now and you let evolution run for 1 billion years, you're saying, well, do I expect that 1 in 100 nucleotides along the sequence will be changed? Do I expect 2 out of 100 or a half out of 100? That's what mu is, okay? It's what fraction of the sites will you expect to change. Another way to think about it is over your period of evolution, as you play God, let's suppose you're going to simulate 1 billion years with some poor organism, how many times do you expect that site to change? Is it once? Is it 10? Is it an eighth? A millionth? That's what mu is, right? Okay? So I prefer to think of it as like out of 100 nucleotides, how many nucleotides do I expect to change over the period of evolution that I'm interested in? Okay. Well, that's just this part here. I just copied the equation for simplicity to see. So I created a little script in R. It's available to you in the Lecture 16 project at the cloud. So I just plotted out here now. So here, this is actually, I should write mu. This is mu. Okay. So here I started by saying, well, I expect no changes at the site, right? So if I expect no changes at the site, what is the probability then I'm in state? Okay, so yeah, the red curve is that I stay in the same state, right? I start in state I, and I end in state I. So whatever state I start in, whether it's A, C, G, or T, what is the probability that I end in state A after basically no evolution, when I expect no changes per site? Not surprisingly, it's 100%, right? Okay, probability is 1. It's a sure thing, because if I don't expect any changes, then I'm in state A, so I stayed. So likewise, at the same time, if I don't allow any evolution, right? And if I start in state A, I'm not going to end in a different state J, right? Okay, so this guy here is this, right? And this guy here is this equation here, right? Just to be clear. I hope that's 100% clear, right? All right, so there's no probability to switch away from state A if there's no evolution. If I expect that site to change once over my period of evolution, well, it says that my probability of being in state A is around, what is that? Looks like Georgia at about 94% of the votes counted, about 20%. Does that make sense? So here I'm asking, what's the probability that I started in A and I mutated away from A? Well, that would be something like, I have a 75% chance. In that model we talked about before, right? In this model here, let's look at it. That's we're looking at this equation here. It's 25%, 0.25, right? Minus a fraction of 1/4, right? A fraction described by e to the minus 4 mu. So this is minus 4 over 3. So if you ask for something like e to the minus 4 over 3, well, that's going to be probably, well, you can put it into r, but that's going to be less than 1/4 for sure, right? And that's going to get you down to here, okay? Now, if I start to allow more evolution, same thing, I've dropped. If I'm state A, look how much I've dropped already in one. And that's because if I started in state A, the chance that I stay after one evolutionary step drops off pretty dramatically. Now, if you allow more and more changes per that site, what happens is that you're basically in any four of the states randomly. Now remember that the probability in the Jukes-Cantor of any nucleotide is 1/4, okay? So it doesn't allow for nucleotides to have different probabilities, right? And that's, let me make sure, a few slides ago, I just want to make sure that in the Jukes, this is this guy here, right? This vector here. So that now I want to revisit that discussion, because I said it's like this pi, our initial probabilities, and I gave this this thought experiment where you pick a site in the yeast genome, play God, and let it evolve for 1 billion years, then look at it and ask what's the probability that you're in state A, G, C, or T. Now maybe you see a little bit better what I was trying to talk about, describe. Basically, after you've had a significant amount of evolution, the probability that any nucleotide is or isn't what we started at approaches 25%. In other words, it's throwing a four-sided coin, right? Or maybe four-sided die is a better phrase than a coin. But you get the idea that these probabilities quickly converge on just random, right, at any given site. Okay. I wanted to basically come back here and tie this now to phylogeny, which is where we're going with this, and in next lecture too. So often we're looking at one species A at some site, maybe some nucleotide in a gene or in the genome, and we're looking at that same, we can call it orthologous site in another species B. And so this up here is the least common ancestor of these two organisms. So it might be, for example, if A and B, if these were both, this is a bacteria and this was, human, then this guy here represents the evolutionary point where bacteria broke off from humans, right, or from eukaryota. So this could be a very long period of time. And this is the period of evolution, this plus this, that separates those two species. And so that the total length of that red line is the amount of evolution. In other words, and that's going to be proportional to the number of times you've walked through your your Markov chain has changed states, right? So this guy here, this might be my organism A, and this might be my organism B, and this red curve that is basically the sum, well, of all of that red line, right? That goes from that red line that goes from here to here, that's this time along this axis, right? Okay, hopefully that's clear. I should get rid of this guy here. There's no confusion. So what can happen? I mean, presumably, okay, so this is now, I should change my marker color. This in species A is a C, okay? And this is a C in species B. Now, somewhere along this point is this guy, this point here. Let's call it star, right? So, you know, we could think of it in different ways, but here the arrow time is going this way, and we're sort of moving from upward from one leaf to the root, and then down from the root back to C, right? So I'm playing with time here, right? The reality is that time is something like this, where the progenitor, I'm going to use a different color here, just a pink or something. The progenitor was in some state, I don't know, maybe make it a bit bigger. We can fill that in later, but we don't know where it was. But then basically it evolved, right, to state C. That's along this arc here, okay? And it evolved towards B this way, and that's this guy as we move along this arc here, right? So this arc, this series of arcs, right, this path is this path here, and this path is this path here. Now, lots of things could have happened. It may the simplest, most parsimonious maybe explanation, we could use that term, would be that this was a C, and it stayed C all the way along in both directions. But of course, that doesn't need to be the case, of course, because it could have had a very complicated history. It could have went from C to T to G to C, right? And it could have, gone to A to G to C, right? So it could have bounced around in that Markov chain, and it's just dumb luck that it ended up back at C at these two children, right, down here in species A and B. Now, which one is the correct explanation? Is it the one where it was C the whole time? Or is it one where it bounced around many, many times across the evolution? I don't know. But that certainly has something to do with the total length of evolution, right? It has something to do with the length of this whole path. In other words, if we go back to common sense approach, view of this, it must have to, if you were comparing Saccharomyces cerevisiae to another yeast that's only diverged by 1 million years, that's one thing. If you're comparing, that means that the total length of this red path is probably, quite short. It's 1 million years of evolution. That's certainly different than if I was comparing Saccharomyces to human and going back to the progenitor where bacteria separated from eukaryotic. That's billions, probably billions, or at least hundreds of millions of years of evolution. Billions. So that almost seems unlikely that nucleotide has never switched state from C. And I would probably think that this is the correct model here, right? Well, this is the basis of molecular phylogeny, and it's the basis of sequence alignment. So we're going to come back to that. I'm trying to foreshadow here of how these techniques are used. Okay, so as I alluded to before, a lot of these models are give the rate matrix, this Q, in this equation, right? And we, because we're all Markov model experts now, we think in terms of these probability transition matrices of the Markov model itself, but that works its way into this equation, right? That the probability of in state, some state at time t plus this unit delta t, a small unit of time delta t, is equal to the probability of in that state at time t plus this equation where it's q times that time interval. And this q for the Jukes-Cantor can be written as follows. It's z over 4 everywhere here. I should, so there's a little bit of a, I don't know what these points don't mean anything, guys. Okay, so it's z over 4, and that's related back to this mu as basically mu is equal to z over 4, 3z over 4, just z over 4 times 3 times the time interval, okay, t. So, well, that's not so important. It's just that, well, I guess there's two things here, is that I want you to understand. One is that for whatever reason, statistical geneticists express these models in this form, this rate matrix, okay? And although I want, and that's fine, because when you're reading textbooks, you understand that. But this is really the Markov model that sits underneath that Jukes-Cantor model, okay? So I want you to give you some intuition of where they come from. And recall, what this matrix Q does is kind of magic, right? It's some heavy mathematics, but if we just go back to this, you're looking at all possible ways of going from what happens starting in state A and how you end up at state A at time across this delta t, and that's a continuous time Markov model, and that's certainly outside the course, but that Q sums up all of those paths essentially. Okay. All right. Well, that Jukes-Cantor is a famous first step. The Kimura 1980 model is a slight modification of Jukes-Cantor, but now it has two parameters. One that controls the probability of transitions, and one that controls the probability of transversions. Okay, so it's like the Jukes-Cantor, all nucleotides are equally frequent. So this pi A is equal to pi C, etc. That doesn't change, okay? But, okay, what's a transition? So transitions are the orange arcs. So A to G and C to D are transitions, and the transversions here are in green, okay? So, yeah, there's more transversions than transitions. Well, chemically, do I have a figure here? Oh, I didn't put them in, I'm sorry. But basically the rationale of this approach is that transitions are more common than transversions. Chemically, A and G and C are more similar to each other than A is with C or with T. And likewise, C and T have the same, the actual nucleic acid, when you look at the chemical structure, are very close. And so basically, when mistakes are made copying our genomes, A tends to get replaced by G more often than A would be replaced by a T or a C, and that's true for symmetrically for the other guys, okay? And so basically what that means is that transversions are usually have lower probability than transitions. So these arcs have higher weight than these green arcs, okay? The orange have higher weight than the green arcs. And so the Kimura model looks like this, ACG and T, ACG and T. And we have two things, one and a K or a kappa. Usually kappa is the notation, but it doesn't matter. I don't like to introduce Greek letters if you don't need to. Here, basically, the idea is that you set transversions to be 1, and then you set the rate of transitions, this parameter K, relative to this. So, actually you'll notice that there's only one parameter in this model, which is K, which seems like I'm lying. But the point is that it's arbitrary. I could set this to be 2, then I would just set this K to be 2 times K, right? I would just update it to be 2 times K. And if I wanted this guy here not to be 1 but a half, then I would set K instead to be K over 2, right? I would just have it. So there's no reason to have 2 separate parameters. One, you just by convention say that transversions is 1, and then calculate the ratio for K. Okay, so the point here, I think, is that we're integrating mathematically more biology into this model by considering basically the chemical reality of nucleic acids. Felsenstein went in a different direction in 81, and it's an extension, should be extension of JC, but yeah, it's also an extension of the Jukes-Cantor 69, but it allows for non-equal probabilities of nucleotides, which is, we know, right, from your own exploration of the yeast genome is true. So now, basically, pi A, pi C, G, and T can all be their own separate values. And so the rate transition matrix looks very simple, actually, in the Felsenstein model. You just put the probabilities for those nucleic acids in the matrix, and it works out very simple. And of course, this is going to be equal to 1 minus the sum of these guys, right? As we know. Oh no, sorry, it's not 1 minus. I'm sorry. It's not, right? It's negative. That would be right. I just made the mistake. This is not a probability transition matrix of a Markov chain. It's the rate matrix, and so this is actually minus the sum of pi C, G, and T. Okay, but that's also, again, one step forward from the JC model, because it also brings in the reality that nucleic acids don't show equifrequently in our genome. Finally, you have the HKY, Hasegawa, Kishino, Yano, 1985 model, and it combines the Kimura and Felsenstein model, so I thought I should show that one. Basically, now we have two parameters. Notice that Felsenstein, you can't say that transitions and transversions have different probabilities. It's back to Jukes-Cantor that way. Here now, I've just added this kappa, or K, for A to G. And A to G, recall, is a transition, okay, like here, A to G. And all the transversions, like A to C, they stay with just pi, right? They have a 1 there. But all the transitions now get a K in front of them. For example, C to T is also your transition on this side. So easy peasy. And again, this is your rate matrix for the Hasegawa, Kishino, Yano. And you can figure out the asterisks easily. In fact, there's an awful lot of different models for molecular evolution. I've only shown a couple. And this table kind of summarizes it nicely. Can you have differences in base composition for Jukes-Cantor? No. It's all equally variable, but like the Felsenstein model is variable, right? And, we looked at one model where the transversion rates were different, et cetera. And they sort of, build up. We stopped at this guy here, but there's other possible models out there for modeling nucleic acids. All right, so far we've been looking at models of nucleic acid evolution and for SNPs, or single nucleotide polymorphisms. And a single nucleotide polymorphism is just a mutation from T to A or A to C, whatever, between two nucleic acids. The nucleic acid changes. And really the models we've been looking at really apply to junk DNA. And there's a lot of junk DNA in the human genome, although that's an extremely controversial topic right now. I put it's not required, but it's recommended reading in lecture 16 from a famous molecular evolution researcher, Ford Doolittle at Dalhousie University. it's his rebuttal to an international consortium called ENCODE that sequenced very deeply and analyzed very precisely the different fragments of the human genome. So that consortium called ENCODE, they went in, they didn't look at the entire human genome, they just looked at certain predetermined areas that were tractable, not too large. And they sequenced them, and they did all sorts of studies with, like, ChIP-seq, or maybe back in the day ChIP-chip, but these are studies for trying to figure out where binding sites are, etc. And well, it seems that maybe we think that a lot of our genome, like 98% of it, for example, is just junk, but ENCODE is suggesting that's not the case, and they presented evidence that I think that almost 80% of our genome has some sort of functional significance. That definitely goes against sort of the classic notion of junk DNA, and that our genomes have parts that don't control don't have any regulatory or functional significance. So that back in about 2010 or so was very hotly debated, and it's a nice paper to read. But for our purposes here, we don't need to worry about that too much. There definitely are sites that are not under strong evolutionary constraints, and that's important, because then they don't play significant roles during selection, right? So if some nucleotide in the genome has nothing to do with the fitness of the individual, for example, in any way, shape, or form, it doesn't code for a gene or a non-coding RNA. It doesn't code for, sorry, I meant to say a protein, or a non-coding RNA. It doesn't have it's not part of a binding site or an enhancer or anything interesting. Then when it mutates, nothing happens, right? And the models we've looked at are really about those kinds of sites. And it's often said that they generally mutate in a clock-like way. It means the mutations come in, and I don't know, whatever the speed of that Markov model is, right? If you understand what I'm saying, you think of God or evolution as being a Markov model, it just ticks away, right? And it'll just flip from A to C to G back to A to T, and go through a kind of a random walk through that Markov model. But if you're in a coding region, for example, well, more generally, if you're not in junk DNA, if you're part of a binding site, or you're part of a non-coding RNA, something that's functional, that gets transcribed or is bound by something like a transcription factor, when it mutates, it may have an effect on the fitness of the individual, and so it may be selected out, right? The organism may not survive. Okay, so it's deleterious, right? Okay, so oops, this should not be axons, it should be, and when you're in an exon, of course, you're then part of the genetic code. So your nucleic acid at any site then is part of a codon, and that codon codes for a specific amino acid. So, for example, if I'm standing at the nucleotide U, and the next nucleotide is a U, and the last, the third letter is a G, then that codes for the amino acid leucine, right? And I guess all of you know the genetic code by heart. Okay, so now some mutations will be synonymous and some won't. Some will be non-synonymous. So what does a synonymous mutation look like? Well, if my genome is U, U, G, okay, then this gets translated as leucine, okay? Now, if this position is mutated to, I don't know, let's say an A, okay? If it gets mutated to an A, it still codes for leucine. So it's synonymous, right? Those two codons code for the same amino acid. Now, you could think that that won't have very much effect on the organism, right? Because the amino acid, which is, that amino acid chain is what folds into a protein, hasn't changed at all. Okay. However, if I go back here, and instead, okay, I mutate. Where's my blue? If I instead mutate my first amino acid, nucleic acid, I'm sorry, to an A, I now have a UG, which codes for thioethene, right? Okay. Now you've changed the amino acid, so that's a non-synonymous mutation, and potentially when that amino acid chain, that polypeptide, folds into a protein, you may change the functionality of that protein. And if you change the functionality of that protein, you may change the fitness of the individual. We don't usually choose to model coding regions at the nucleic acid level. Instead, we use amino acids at that level, so the translated nucleic acid sequence. Okay, now amino acids certainly have different physical and chemical properties, and we can split them by like their charge side change, positive, negative. You can split it by the polar side chains. You can special cases, and then finally hydrophobic side chains, right? Hydrophilic. That's really, this guy here is really important because hydrophobic amino acids tend to be in the core of the protein. They get folded and they're hidden from the surface where there's water. Example, you have your hydrophilic amino acids on the surface. Okay, and yeah, and then of course these different amino acids, their different properties and charge are what confer them certain properties. Let's say in active sites and binding and things like that. Okay. Well, in the required reading, I left a couple links for describing how amino acid chains fold to protein structures, and I won't go into that here. The Smart Biology is really nice. I love those textbooks. And there's another video from YouTube that describes the basic principles for those people in the class that haven't had a course in that direction yet. It gives you the intuition of how the amino acids, changes in amino acids can change protein function, structure, etc. Yeah, point accepted mutations, PAM. So PAM matrices were actually built by a woman named Margaret Dayhoff back in 1978. She's pretty famous, actually. She's pretty visionary. And she collected about 1,500 mutations at the time. So what she did was collected, I think, 70 phylogenetic trees, or 71 or something like that, that were made across different protein families, like ribosomal subunits and, I don't know, essential genes that crossed a lot of different I think these were all eukaryotic organisms, human, mouse, chimp, etc. 1978, there wasn't a lot of data out there, but she put them all together in these trees, and she observed basically that there were 1,500 mutations across the tree. So actually, I don't have a slide from her paper, but basically, just I don't want to make you dizzy here, but to go back to this figure, she built these phylogenetic trees for different proteins, and then she looked at the leaves and said, okay, species A and species B have a mutation at some site like this, right? Okay, and she found over 70 such trees. She found about 1,500 such mutations, so different sites. Of course, she's looking at the amino acid level, not at the nucleic acid level. Okay, now a PAM matrix is a 20 by 20 matrix related to the probability that amino acid Y transformed to amino acid X. Yeah, they're also derived from a Markov model. I'm going to spare you the mathematics there, but definitely there's a Markov model underneath the PAM matrices. And you can see the PAM matrices in R. The biostrings library, which you have already been using. If you do data, you'll see PAM 30, PAM 120, PAM 250. It has a bunch of different PAMs, and you can view them with the view function, PAM 30. So a PAM sub I matrix, whether it's PAM 1 or PAM 2, PAM 2.5, it can be fractional, but it represents the probabilities for amino acid transitions when we expect I mutations per 100 amino acids. So this is the measure of evolution now. It's a much like that mu parameter we saw before with nucleic acids. So the same thing. You can see it in different ways. You can think of it as like, okay, over my period of time, if I have 100 amino acids, how many do I think are going to be mutated in that time? Or you can think, I'm looking at one site, right? And over my period of time that I'm interested, from t to t plus till delta t, that's time now, right? How many times do I expect that site to be hit, right? Okay. So that's our unit of evolution. And it goes from PAM 1 to PAM 250. Now, they're continuous Markov chains, so we can actually reason about PAM 2.5, but that's irrelevant, and you never need to think about that again for the rest of your life unless you somehow end up as a mathematician or biostatistician and you're interested in that. So PAM 250 is actually really important because it represents the mathematical limit of detecting sequence similarity. This is like 250 iterations of the Markov model, right, in essence. And so it's saying that we expect 250 mutations. I take that back. It's not 250 iterations of the Markov model, but it's a lot of iterations of the Markov model until we expect 250 mutations for every 100 amino acids. Okay, so it's a lot of evolution. So how PAM matrices are derived from these from a Markov model is well beyond the scope of this course. I'm not going to torture you with that, but the intuition is pretty straightforward. So like Dehoff, we can start with a set of examples of alignments of proteins from very closely related species. Let's suppose that species A and B, we would think of them, we wouldn't be comparing yeast to human here. This would be like rat to Trump, or yeast, baker's yeast to Espombe, human to monkey type thing. Okay, so actually we would want these two sequences to be aligned, which is the subject of next lecture. But intuitively, if we believe that this is a fragment, a peptide fragment of a protein from species A, we find the same protein in species B, and we match them up. So isoleucine, leucine, proline versus tryptophan, et cetera, okay? And here we actually observe a mutation, right? We see here a point mutation between these two sequences, right? And it's been accepted. It's been accepted. We say it's been accepted because we wouldn't, if it weren't species B or species A, we're not sure which one. We don't know at the root whether it was a proline or tryptophan, but we do know that both species survived, so they can both survive with either a proline or a tryptophan in that position. So let me draw something out here. Okay, I change my color code. I'm going to go over to, let's say, a green. Okay, no, that's not neutral. Here, let me do this. Blue. So this is species A and this is species B, and they both derive from some common ancestor, right? And at species A we have a what is it, a P, a proline, and at species B we have a W, right? So we don't know what was up here. Maybe it was a proline, okay, and it stayed a proline the whole way, and it switched somewhere along this branch. It switched from proline to tryptophan, or vice versa. Maybe this was a tryptophan, and it switched along this arc to a proline. Or a third possibility is, of course, that it was something entirely else. It could have been, arginine that mutated to a proline along here and mutated to a tryptophan along there. It doesn't really matter. The point is that whenever this switched to a proline or whenever this switched to a tryptophan, whatever occurred, wasn't so deleterious, it wasn't so negative that both species could survive. Okay? And that's important because it's not captured by our previous models. Here we have selection. Because the mutation, ultimately the mutations are at the nucleic acid level, but since some of them are non-synonymous, some of them are synonymous, they don't have any effect on the amino acid and therefore the protein and therefore the function. But some do. In this case here, there must have been a nucleic acid mutation that switched from proline to tryptophan somewhere along the line, and that potentially changed the function, or let's say the fidelity or the ability of that particular protein. But the mutation didn't kill the organism, otherwise we wouldn't see species A and B. But with Margaret, what she did day off was she was collecting lots of these fragments of across 71 trees, and she collected a lot of these fragments that you see here, 1, 2, and whatever, as many as possible. Like now you would use hundreds of thousands, right? But back in the day she didn't have too many, just 1,500. And then she calculated the frequency of each amino acid, which was just like the number of times she observed the amino acid divided by the total number of observations. So here it'd be like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So big N would be 13, right? Because there's, plus all these guys, right? It's the total number of sites. And this guy here is just the total number of times, for example, you see an I. So 1 in this example here, 1 out of 13, right? That's pretty straightforward, I would imagine, for everybody. But then, so we use the examples of mutations and non-mutations in the learning set to build a transition probability matrix P1. And so we think of P1 as the probability of going from amino acid to amino Y to X. I've always hated this, that somehow they reverse it. I always think of X going to Y, and in all of our Markov chains, that's what we are doing, is X to Y. But in PAM matrices, it's reversed. I really don't like it, but that's the convention. So it's amino acid to amino acid in one step. So think of that as one step of the Markov model, okay? And P2 is the probability of going from Y to X in 2 steps of your Markov model. And 250 is the probability of going from Y to X in 250 steps. So more formally, our PAM at N, whatever that is for IJ, is this log ratio that you've seen before as the background distribution and transcription factor binding sites. And here, the numerator is that is the frequency of J. So you started in amino acid J, right? And that's this guy. And you transited over N steps of the Markov chain from J into I. So think of this as like simulating that Markov walk for N steps and ending up in state I. Okay? Now, that's divided by the background, which is just the probability of J, right, occurring in random, right? And not all I should have had a table in here. It's not the case that amino acids show up equally random. There's a big difference. Some amino acids like tryptophan are very rare, and other ones like isoleucine and leucine alanine are very common. Like 20% of amino acids are like leucine, I believe. Don't quote me on that, and I think less than 1% or 1% are tryptophan. So they have very different frequencies. But this is the frequency that you find something in state in amino acid J times the frequency that it switches to I. So what's I think you should stop the video then and ask yourself, what's the intuition? Why do you take the log of this, right? So this is the log of the probability derived by the model divided by the probability of just, what we observe being kind of random, right? So think about that, and we can talk about it in class. So in the end, the PAM1 matrices represent transition probabilities for closely related species. It means, like, you think about it, it's like if I have two really close species like human and chimp, even closer maybe than that, but you think about, it wouldn't take very many steps in the Markov model to convert me into a chimp, right? But to convert me into a yeast, you would really have to change a lot of things, right? And that might take hundreds of steps in that Markov model. So so it's quite simple. The fewer the steps, the closer the species. The larger number of steps, the larger the species. The larger the distance between the species. So I put some examples here that you could work through. I'd like you to try to understand them. Now, the code for looking at these matrices is in RStudio. I've shown you a simple alignment, so T versus TE. There's a mutation here, a mutation there, and a mutation there, and a mutation here. And this is for PAM30. And I'll let you work through and see if you understand why I get the score of 9. And then I show the same, but I've changed the PAM matrix now to 120, and I just get a score of 13. And finally, I changed the PAM matrix to 250, so a lot of evolution, and I get a score of 11. So I want you to think about this. Which was the right matrix to use for those two protein sequences? Okay, what would be the rationale for choosing the right matrix? Okay, now here I don't know what these two species are, A and B, but what would you say here? What would be the rationale? The second thing I'd like you to look at is the diagonal of these matrices. Now this is PAM30, and when you look at PAM, maybe I can zoom in a little bit. You'll notice here on the diagonal of this matrix that the values are all very positive. And the off diagonals are extremely negative, like minus 17. You can try and find the smallest one you can find, but they're quite negative. Now, when I go out to PAM 250, this one, you can see that the diagonals are still positive, but they're closer to 0. And the off diagonals are often negative, but not always. There's actually some positive values and zeros. So for the class, I want you to think about why that's the case. So succinctly, why are the diagonals in the low PAM matrices very high, but they're closer to 0 at the high PAM, like at PAM 250? And why is the opposite pattern seen for the off diagonals? Okay, so we're going to stop there. Okay, actually, I'm going to make one final statement to say that there's another type of matrix that's called Blossom, and they came after the PAM matrices, and they're considered to be very good. Now, their methods and the Markov models, etc., are very similar. Their input, the way that they process their input for calculating their matrices was a bit superior to the way the PAM matrices were formed. And there's some other minor differences, but often in practice, Blossom matrices are considered to be an improvement on PAM. I show the PAM here because it's the concept that really counts of where these matrices come from. We're going to pick up there next class.